Okay, folks, so here we are on the fourth and final portion of the Relationships in Biodiversity Lab. I know that part 3 was a little on the longer side, so I apologize for that. Hopefully this one won't be quite as long. I am going to ask that you do some of this on your own because it is going to be a little bit dependent on your results and on your data chart. So remember that this is in addition to activity number 9. So in your classroom, of course, again, I'm going to the pink classroom. Doesn't really matter if you're pink or green. It should be the same. You're going to go down to your ecology section. You should have number 9, relationships and biodiversity. Obviously, again, yours looks different than mine, but you're going to open up your document, which should be here. At this point, you should have filled in, give that a second to load. You should have filled in, page 1 is nothing to fill in. Page 2 should be all filled in now because there's only 2 things you had to fill in. That should have been done in video number 1, I think. Page 3 is all instructions. Page 4 should have been done in video number 3, in the last video you just watched. Not sure why it's not loading here, but this is the part on the bottom where you have to translate the DNA into RNA, and there we go, and then translate the RNA into amino acids using the amino acid chart. So page 5 and 6 are what we're, and 7, sorry, are what we're going to do today. Page 8 is the data chart. That should be all done. So this should be all filled in from video number 3, and page 9 also was a part of one of the tests, so that should already be filled in. It's probably highlighted. If you remember, you had to click in the boxes and then click on fill color, and you could highlight little banding patterns. So together we're going to start on page 5. Zoom in a little bit. Some of the answers I'm going to type with you and others I'm not. So the first question here says, state how the amino acid sequence you obtained from the gene fragment from Botanicurus compares to the sequence for the other 3 species. So that's based on this. You're looking at the amino acid sequence, which you should have in the second row here, 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 and here. You can also use, where is it? You can also use your data chart. The differences in amino acid is in column 7, which is this column with the gray box at the top. So all you have to do is talk about what's different and what's not different. Since I am at home and I don't have my answer key with me and I didn't do these all the way through, I'm going to ask that you answer that question there on your own because I don't really have a great example of what to give you. Okay? So go ahead and if you need to, pause the video and you can answer this question on your own. You're basically, again, writing what you have in the data chart for column 7, what the differences are between Botanicurus, which is the top one, and then species X, Y, and Z. So then if we were to analyze the results, using the information in table 1, identify which plant is most closely related to Botanicurus and therefore more likely to produce curel. So again, you're going to use your data from page 8, from page 8, and you are going to type right here. You can highlight this and delete it, of course. You're going to type in there and say which species, X, Y, or Z, do you believe is most closely related to Botanicurus and therefore most likely to produce curel? Explain your choice by citing specific evidence from your research. So here, what you need to type in, so you're going to put your X, Y, or Z right here. In this question, you need to type in specific evidence from the data chart. So go to your data chart on page 8 and say, well, in test number 1, we noticed that both Botanicurus and species, whichever, have blah, 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 whatever characteristic that you noted in tests 1, 2, and 3. Or you might say, well, when we tested for enzyme M, both species, or both Botanicurus and species Z, if that happens to be the one you picked, they both are positive for a test for enzyme M. Or maybe a different one, maybe the gel electrophoresis banding pattern for Botanicurus and X is the same. Maybe it's not, I don't know. But you have to decide, and you're going to put that for your evidence here for question number 1. So here in this box for question number 1 on page 5, you're typing in which species X, Y, or Z, and here you are giving specific pieces of evidence why. I would think that you should have at least 3 pieces of evidence. You have 7 tests, so pick 3 pieces of evidence, 3 tests, and just give me like a little bullet on each. You can just do a bullet for each one. Okay. So number 2 says, did the addition of molecular evidence support or refute the hypothesis that you made earlier based on the structural evidence only? So now we're asking you to go back to page 2, which looks like this. Up here you should have already put X, Y, or Z as your prediction. You are referring back to that. That's your prediction. So according to number 2, did the addition of molecular evidence, which would be tests 4, 5, 6, and 7, refute the hypothesis you made earlier based on the support or refute the hypothesis you made earlier based on structural evidence only. Click here to type. Okay, so you're going to click here and say yes or no. Did it help? Or I'm sorry, you're not saying yes or no. So did the addition of molecular evidence support or refute? So did it support it, which did it continue to make you think that whichever you predicted up on page 2 is in fact the most closely related to Botanicurus? Or when we added in those last 4 tests for the molecular evidence, when we tested for enzyme M, we looked at the amino acids, when we looked at the chromatography, and when we did the gel electrophoresis, did those things then make you think, wait, no, what I thought on page 2 is not right? That means it was refuted. So either here you're going to write supported or refuted. If you think it was still what you originally thought, then you're going to put supported. If you now think it's something different, so if what you wrote here for number 1 is different from what you wrote up here for page 2, then you're going to write refuted. Okay, and again, explain why or why not. You want to use specific evidence from your data chart on page 8, okay? At least 2 to 3 pieces of evidence here. Again, you can do them in bullet form or do a dash and just a quick blurb. Doesn't have to be a full paragraph, but you need to give at least 2 to 3 pieces of evidence. So go ahead and pause the video if you need to, and you can answer number 2. Number 3. Which kinds of evidence, structural or molecular? So remember, structural is test 1, 2, and 3. Molecular is test 4, 5, 6, and 7. Which kind of evidence, information we got from the structural test or information we got from the molecular test, is the most helpful in making decisions about relationships? Which one do you think? You're going to click here and say either structural or molecular. Okay, so think about it. When we looked at the structural evidence, we were looking at the external characteristics. We were looking at the number of petals, maybe the color of the petals, the types of seeds, the shapes of the seeds. I kind of tried my very best to show you the test 3 with the little microscope slides. But all of those different things, are those things more helpful in making a decision about the relationship? Or are things like looking at DNA, looking at amino acid structure, or looking at paper chromatography more helpful? So typically we would say that molecular evidence is more helpful. Molecular evidence is more helpful. And we would say that because what does molecular evidence give us? Molecular evidence is evidence that is based on internal structure, such as DNA. So the gel electrophoresis, that comes from DNA. The amino acids sequence, that comes from DNA. If you remember your 5 fun facts, remember that DNA is the code or the recipe to make proteins, and proteins are made up of amino acids. Okay, so the information from the DNA gets sent over to the ribosome to create the protein. And if the DNA says one recipe, then it's going to make one particular protein, where if it says a different recipe, it's going to make a different protein. So evidence that is based on internal structure, such as DNA, and maybe enzyme reactivity, will be more helpful in making decisions about relationships between species This is because DNA is much more, let's say, change that, sorry. This is because molecular evidence, not necessarily just DNA, but even the paper chromatography, which was looking at plant pigments. This is because molecular evidence is more specific than structural evidence. For example, if I asked you to describe characteristics of me and characteristics of Cooper, and I said, well, I have 2 ears, and so does Cooper. And I have a nose, and so does Cooper. And I have a mouth, and so does Cooper. And I have 2 eyes, and so does Cooper. If I had all of those things, and so does Cooper, does that mean we're definitely related? Well, if you looked at a picture of us, you would know that Cooper is in fact a dog, and I am a human. So we are, sure, eventually, somewhere along the evolutionary line, maybe, who knows, but we are not very closely related because we're different species entirely. We're different organisms. So you could ultimately end up looking at 2 different plants that happen to have similar characteristics on the outside, and those outside characteristics may not be necessarily telling you much. So again, like the dogs, most of the pets you may have in your house would also probably have 2 eyes. I have fish here too. They also have 2 eyes. Does that mean I'm related to the fish? No, it does not. Just because we both have 2 eyes doesn't necessarily mean we're related. We would want to look at our molecular structure, our DNA, our enzyme reactivity, and things like that to really know for sure. Of course, most people would know that you are not very closely related to fish, nor am I very closely related to Cooper. Okay. Question number 4. Question number 4 says, based on your observations, list 3 characteristics, structural or molecular, that all 4 species have in common. So here you're going to do this one on your own. 3 characteristics. You could do it in bullet form, and it has to be something that all 4 species have in common. Okay? You might find that all 4 species have more structural things in common than all 4 species have molecular things in common. So I would give you a hint and encourage you to start with tests 1, 2, and 3. But you might find that you have similarities in test 4, 5, 6, and 7 as well. Okay, so 3 characteristics, structural or molecular, that all 4 species have in common. You're going to list those here. And again, you can just do bullet points. So go ahead and pause the video if you need to. And you can do that part. Okay, so the next page, it says, provide a biological explanation for the common characteristics that these species share. Why do they share common characteristics? Why do I share common characteristics with you, or with my mom, or with other humans? Why is that? Well, likely these species share common characteristics because they are in fact related. Some of them, they probably also have the same or similar DNA structure. They all have a similar DNA structure, but based on the molecular evidence, they must not have identical. Yep, I spelled that right. Identical DNA. So remember, that's a big distinction between similar versus identical. Okay, so that can be a big distinction. It's important to understand the difference there. Obviously, similar is similar. My mom and I have similar DNA. Even humans across the board have similar DNA. However, your DNA is not identical to unless you happen to have an identical twin, of course. But your DNA is not identical to your relatives, to your friends, to your classmates, to your teachers, to your parents, to those kinds of things. Your DNA is not going to be identical to those things. To those people, unless of course you happen to have a identical twin, then your DNA probably is identical. The next one says, scientists frequently use branching tree diagrams to represent graphically the relationship between species. Which branching tree shown below best represents the relationship among the 4 species? So I will tell you now, after you have gone through this, that your answer above, on number 1, should have been that species Z is the most closely related to Botanicurus. So hopefully, based on the other videos that you've seen and the observations that you've made, that was your final conclusion. So again, that's not to say that the other plants are not related and that they're not similar, but we think that species Z is the one that is the most closely related. So if you remember back to evolution, think about this is your beginning and it's kind of like a timeline. So if you think, it doesn't necessarily mean or matter how long ago the time is, but it's just a long time ago here, and this is moving towards the present. So all of these frontline organisms, which is all of them because they're all still living, are continuing to live through the present, if you remember how to read your evolutionary trees. So we have determined that species Z is the most closely related to Botanicurus, and so which evolutionary tree will show that, which I realize means I'm supposed to write one of the evolutionary trees there. Sorry about that. Which branching tree shown below best represents the relationship among the 4 species? So if you know that Z and BC are the most closely related, then that means they have the most recent common ancestor if you're moving from a long, long time ago in a lateral movement towards the present. If you were to draw a timeline from here, these numbers down here, all the way to the front, or the top, I guess you could say, then it would be hopefully clear that the most closely related, or the tree that shows that Z is the most closely related, would be number 2. Okay, so again, all 4 of these species are still alive. They are still related because here is their all 4 common ancestor. But Z and BC have a more recent common ancestor. Again, if you think about a timeline moving forward, this is much further back in history than this point right here. And so that is why we say that it's number 2, because it is most closely related to the Z, even though it is in fact probably related to X and Y. Explain how you use the information in the data table to select this tree. So now you're going to kind of justify your answer, again, based on the data table on page 8. So I'm going to let you do this part on your own. You're going to use the data table on page 8. If you need to go ahead and pause the video, you're going to come up with specific evidence that shows, again, why BC and species Z are the most closely related. Hint, if you didn't think that in the beginning, you may want to go back and revisit. Maybe you need to go back to video 3 and revisit that a little bit. Otherwise, I would just at least go back to page 5 here and revisit some of these questions before you move on. Because it is Z. Don't tell anybody, but it's Z. Okay, so go ahead and pause the video if you need to and answer the rest of number 6. Number 7 says, state 2 additional kinds of evidence you might use to further support your hypothesis about the relationship between Botanicurus and species X, Y, and Z. So this is one that people really struggle with. This is one that even I really struggle with sometimes. But here's a couple of ideas. So 2 additional kinds of evidence you might use to further support your hypothesis that the relationship between Botanicurus and species X, Y, and Z. So we already said that we looked at 7 different things. We looked at 3 different types of structural characteristics. We also looked at 4 different types of molecular evidence. So the other thing that I always have kids do is think about maybe where do these organisms live? What else could you think about? This says 2 additional types of evidence. So where do these organisms live? How much sunlight do these organisms thrive under? And again, thrive means to do well. So do they need a lot of sunlight? Do they not need a lot of sunlight? Do they not do really well? Maybe they do poorly when there's too much sunlight. Who knows? And this kind of goes along with, here's another one too. What temperature do they grow best at? Okay, so those are a lot of those kind of fall into this where do organisms live category. We're talking about like their habitat, but that is this question I always find is a little tricky, so that is usually the route that I have my students go. The next page and the last page is a little bit of a reading. It is a biodiversity crisis. So it says plant and animal species are being lost at a rate that is unprecedented in the history of life. Human activities are responsible for much of this biodiversity crisis. Some biologists estimate that within this century, half of the Earth's current species may become extinct. Extinction is the loss of biodiversity. Extinction and the loss of biodiversity occurs when species do not have adaptations that enable them to survive environmental changes. Human activities such as destruction of natural habitats and pollution are thought to be the major environmental factors causing the decline of species, but others are also important. Overhunting, introduction of foreign species that compete with the native species, and removal of predators also have played a significant role in endangering some species. Why should we worry about the loss of biodiversity? We depend on many species for food, clothing, shelter, oxygen, soil, fertility. The list goes on and on. Large-scale extinctions of other species may be a warning to us that we are altering the biosphere so rapidly that our species is threatened too. Biodiversity ensures the availability of a rich variety of genetic material that may lead to future agricultural and medical discoveries, having significant value to humankind. Some species have been used as sources for medicine and other useful products. Scientists now use genetic engineering to transfer desirable genes from one species to another. As diversity is lost, potential sources of these genetic materials may be lost with it. Biodiversity can also increase the stability of the ecosystem. Every population is linked directly or indirectly with many others in an ecosystem. Disruptions in the number and types of one species can upset the ecosystem stability. This means that extinction of one species can accelerate the rate of extinction for another species. Okay? Endangered species hold medicinal, agricultural, ecological, commercial, and aesthetic value. They must be protected so that future generations can experience their presence and value. So, assume that the plant you identified as being closely related to Botanicurus grows rapidly, survives in many environments, and produces cure-all. News reports indicate that Botanicurus plants may become extinct unless expensive efforts are made to preserve the species. Members of your research team disagree as to whether or not Botanicurus should be saved. So again, remember from way back at the beginning, Botanicurus is not growing very quickly, and it is it's not extinct yet, but it is definitely getting endangered, so they're running out of it. So it says, state 3 examples of human activities that could endanger Botanicurus. So if we go back to the reading, we can see that here are some human activities. Human activities such as destruction of natural habitats and pollution. There's 2. Okay, so let's put those in. Oops. Destruction, natural habitats, pollution, and what else we got up here? Human activities such as destruction of natural habitats and pollution are thought to be major environmental factors causing the decline of species, but others are also important. Overhunting, introduction of foreign species that compete with native species, and removal of predators also have played a significant role in endangering some species. So we only need one more, so we'll say overhunting. Or in this case, because it's a plant, it might be considered overharvesting, because you don't really hunt plants, but you hunt animals and you harvest plants. Okay. State 3 reasons why it might be important to preserve Botanicurus. So why do we want to keep Botanicurus? Having Botanicurus, the more organisms you have living in an environment, the more biodiversity you have. So What's so great about biodiversity? Well, biodiversity ensures the availability of a rich variety of genetic material. So, if we keep the biodiversity and increase in genetic material in the environment, that may lead to, you can see up here it says that may lead to future agricultural or medical discoveries. And it says we need 3 or 2. Oh, we're going to run out of room on this one. That's okay. Let's see. Some species have been used as sources for medicines and other pesticides. Now use genetic engineering to transfer desirable traits from one species to another. Diversity is lost. Potential sources of these genetic materials will be lost. Biodiversity also increases the stability of an ecosystem. Increases stability of an ecosystem. So it keeps the ecosystem functioning better. And let's see, disruption in the numbers. Disruption in the numbers and types of species can upset the ecosystem stability. This means that extinction of one species can accelerate the extinction of another species. Endangered species hold medicinal, agricultural, ecological, commercial, and aesthetic value. So that would be our last one. Endangered species can hold medicinal, agricultural, ecological, etc. Let's say value. We don't need to write them all, I don't think. We can scooch this a little longer too and fit it all onto one line. Okay, so there's your 3 things there. Okay, the last thing says state 2 arguments what people might make for not preserving Botanicurus. All right, so great. It's got all these things. Well, maybe we don't need it because we just found another organism that is very similar, but grows rapidly and grows rapidly, survives in many environments, and produces cure-all. So why bother saving the Botanicurus if we already have species Z that will do the same thing? Well, it's fine. We don't need to. Okay, so that's one reason. Another argument you might make, which is kind of similar along the same lines, is it could be expensive to save Botanicurus. And it could be a waste of money if you already have another organism to preserve Botanicurus. So if you don't have a lot of money, then maybe it doesn't make sense to spend a lot of money saving Botanicurus when you already have species Z that will kind of do the same things for a lot cheaper. Okay, so at this point you should have all of this lab all filled in. If you did not, go back and watch the other videos. Obviously I wish that you were watching them in order, but if you didn't, you should probably go back and do that. Okay, I hope everybody is doing well, staying safe, staying sane in this junky weather and quarantine life. If you have any questions, let me know, and I hope to see you all soon.